Okay, so in this video, we're going to continue with meiosis 2. So we've already gone through the first phase of meiosis 1, where we went from one diploid into two haploid, but that's not enough because we still got sister chromatids together, and we need to split that in meiosis 2. So let's follow the video along.
Centrioles again duplicate and begin moving to opposite poles of each cell. In prophase 2,
same thing, prophase. You always have condensation. You always have breakdown of the nuclear membrane. You always have spindle fiber forming and moving towards opposite end and reach out to grab onto the kinetochore. All that is the same. It really, and meiosis 2 really resembles mitotic division, mitosis. Okay, meiosis 2 looks a lot like mitosis. Why? Because we're going to split sister chromatids, which is what mitosis does. Okay, so we're going to start in prophase 2 now.
The sister chromatids condense while the spindle starts to form as the nuclear envelope disappears. In prometaphase 2, the sister chromatids attach to the spindle by kinetochore microtubules, with sister chromatids attached to opposite poles.
Okay, that is a lot like mitosis. Sister chromatids attached to opposite poles. Okay, meiosis 1 is the homologous, the tetrad, actually attached to different poles. The homologous was attached to different. Now it's sister chromatids, and that's the same as mitosis, and they're going to be split in the middle. So we went from prophase, right here in the video, there's prometaphase, and then we're going to go into metaphase 2, where they're going to align in the metaplate again.
The spindle aligns the sister chromatids along the metaphase plate during metaphase 2. During anaphase 2,
right, what happens in anaphase is always the same. Something is being pulled on opposite side. In this case, it's sister chromatids. Okay, I want to remind you, meiosis 1, it's homologous chromosome that got split. Here it's the sister chromatids that got split. So very similar to mitosis. Chromosome align on the metaplate and the metaphase plate. Anaphase, that's where we are, anaphase 2, it's sister chromatids that separate from each other.
Sister chromatids separate and individual chromosomes move toward the poles.
Okay, so we are going to then finally see 4 cells, right? We ended meiosis 1 with 2 cells. Now another split, and we're going to get 4 haploid at the end.
The entire process ends with telophase 2, as the chromosomes decondense and the nuclear envelope reforms.
Okay, again, telophase is everything opposite of prophase. Prophase, if you got chromosome condensing, then telophase decondensing. Prophase, if you have nuclear envelope dissolve, telophase reform. Prophase, spindle apparatus form, then telophase, it actually deforms, right? So everything opposite. And what's the most important thing is that now you have 4 sets of daughter chromosomes, right? 4 sets, 1, 2, 3, 4.
Cytokinesis occurs and cleavage furrows separate the 2 daughter cells into 4 haploid daughter cells. The haploid daughter cells will specialize into gametes, either sperm or egg.
So that's how you end meiosis, is you actually become the daughter cells will specialize, or eventually become the sperm or the egg.
These fuse in fertilization to form a zygote, which will grow into a child.
Okay, so let's look at the answer from earlier on. Diploid, 2 of everything. For human, that means 46 chromosome total. And in male or female, male makes meiosis into half of the amount, which is 23. Female, you go from 46. So let's go through the math a little bit, okay? 46 to start. And then you go through synthesis phase, so you double everything. That means you reach 92. And then you go through 2 rounds of division. So 92 divided back down to 46, and then divide again to 23. That's how you get 4 haploid, 423 chromosome cells, okay? And when the sperm, which carries 23, meets an egg that carries 23, then together they bring back a human, which is 46, which is the baby.
The child receives half its chromosomes from its mother and half from its father. Because men and women produce millions of gametes, and the selection of gametes in fertilization is random, this contributes to genetic diversity. This explains why a child isn't identical to either parent. But why aren't siblings identical? The diversity comes from several sources. In prophase 1, non-sister chromatids can exchange DNA through crossing over.
Okay, so it's giving us the final summary of how we get genetic diversity. One thing is prophase 1, crossing over,
increasing the genetic diversity of individual chromatids. In addition to crossing over, in metaphase 1, the pairs of homologous chromosomes align independently along the metaphase plate and sort independently into the daughter cells.
The second thing happens in metaphase, where we got the random assortment of how do you line up? Is it always all the purple on the left, blue on the left, or you got purple, blue, and then all those are different genes potentially from grandmommy or granddaddy, and how they assort, okay? So these are two major key points.
This process, called independent assortment, produces 4 genetically distinct haploid gametes. As the total number of chromosomes increases in an organism, the number of genetically distinct gametes increases by 2 to the nth power. This means an organism with n equals 3 can produce 8 unique gametes. For humans where n equals 23, there are 2 to the 23rd power unique gametes formed, or over 1 million different possible combinations. The combination of independent assortment, crossing over, and the random pairing of gametes during sexual reproduction increases genetic diversity and explains why a child will not look exactly like his or her parents or siblings.
Okay.
Have you ever wondered why children...
So let's stop there. That's the main key points for meiosis, and I'm going to finish off by kind of going through the PowerPoints. So you can use these pictures to kind of follow along and how it's drawn out. So these are the main thing with meiosis 2, where in prophase, then metaphase is lined up, and then anaphase, sister chromatid splits, and then finally telophase, okay? But right here, I am only drawing out one of the daughter cells, right? So you only got 2 in the end, but you can imagine the other one also goes through. So in the end, you should have 4, okay? And the final result is you get 4 cells of haploid sets. And this part is very interesting. This part talks about what happens if your sex cells does not separate correctly, okay? If it's supposed to split, but it did not split correctly, the sister chromatid did not split, then 2 will go into one sex cell and the other sex cells won't have that chromosome. What happens? Well, that's when you're going to see something called Down syndrome. That's when you are going to see an egg that carries 2, because the sister chromatid didn't split. Instead of just 1, it brought 2 to make the baby. To what? To chromosome 21. So if you've heard of trisomy 21, meaning Down syndrome, where did this Down syndrome individual get 3 trisomy, 321 chromosome 21? Well, it could have gotten 2 from mom and 1 from dad, which was normal. Mom should have brought 1, dad brought 1, but somehow something called nondisjunction, failure of chromosome to move to opposite poles happened. So mom actually brought 2, and then now you have genetic imbalance, and that will show up as the baby grows up, okay? And the non-disjunction during meiosis is going to cause a situation called aneuploid gametes. That means this gamete is going to have a missing or extra chromosome. In the case of Down syndrome, it's an extra chromosome 21, okay? And there are other possibilities like Turner syndrome, which means the female does not have XX chromosome. It only have one X because the other cell did not bring any sex chromosome. Then it's missing a chromosome, okay? So unfortunately, these type of things do happen, and it could happen in random, or it could happen as we age, okay? And a lot of times when this happens, it ends in a spontaneous abortion, so miscarriages. It's nothing that you can do about it. It's just part of the nature of when meiosis happened. It did not split correctly. Okay, so the last few slides summarizes the difference between meiosis and mitosis. So definitely look through them, but I've already talked about this early on in the first video that I made. So definitely go back and watch that and make sure you can compare meiosis with mitosis, understand their differences as well as their purposes. Okay, I think that is all for chapter 11.